Welcome to the Her Health Podcast, transforming health for women and girls at risk, where we empower women and girls to take charge of their health and well-being. Join us as we shine a spotlight on issues affecting women and girls' health and explore ways to create positive change. Get ready to be inspired and empowered as we discuss health, advocacy, and empowerment for women and girls at risk. Please be advised that this podcast may cover sensitive topics such as gendered violence. We understand that these discussions can be difficult to listen to. If you feel uncomfortable or need support, we encourage you to prioritize your well-being. Consider exploring our other podcasts if these topics are triggering for you. Resources can be found in the description.
Hello, and welcome to the Her Health Podcast. I'm Alexandria Lazochuk, a master's student in community health sciences, specializing in public and population health at the University of Calgary. I also work at the Child Studies Lab, where we focus on research that supports children's lifelong developmental health. In today's episode, we're featuring a presentation from Ava's webinar series on how our biology adapts to life's challenges through the lens of epigenetics and cognitive neuroscience. The presentation can be viewed on Ava's YouTube channel at avatraining.ca, alongside with other presentations from the series. We're thrilled to feature Dr. Sarah Merrill, a research fellow in the Department of Psychiatry and Human Behavior at Brown University, specializing in developmental psychology and social epigenetics. We also have Dr. Tayler Eaton, who earned her Ph.D. in Cognitive Neuroscience from Cornell University, focusing on the neural and physiological underpinnings of maladaptive emotional memory. Together, they'll explore how adversity shapes us biologically and psychologically, the transgenerational effects of trauma, and opportunities for interventions to promote resilience and well-being. Be sure to check out the link in the bio for timestamps and more details about today's episode. Let's dive into this fascinating discussion.
Today's topic, we are so lucky to have two wonderful guests joining us, Dr. Sarah Merrill and Dr. Tayler Eaton. I will give them both a moment to introduce themselves, but we're very excited to have you here to join us on their talk on biological adaptations to adversity, evidence from our genes and brains. So we'll go ahead and hand it over to you, Sarah and Tayler.
Thank you so much. We really appreciate everyone coming and for you having us here. My name is Dr. Sarah Merrill, and I am a postdoctoral research fellow at the Warren Alpert Medical School at Brown University in the Department of Psychiatry and Human Behavior. And I work in the Stress, Trauma, and Resilience Center, or the STAR Center, here at Brown. And I work on social epigenetics, which you will hear a lot about today. So thank you for having us.
I am Tayler Eaton. I got my doctorate at Cornell in the same department as Sarah Merrill. My focus was on the neuroscience underlying trauma, and currently I'm working as a senior analyst with Mercer Canada. I will be presenting a little bit on cognitive neuroscience and its interrelation with trauma and the body, and then I'm going to pass it on to Sarah, who will talk about some really, really fascinating work that she's been doing with genetics. So I'm going to start off with just some basic definitions so that we're all on the same page. We're going to be talking a lot today about adversity. Adversity isn't just regular life stressors. You know, if you have a really tight work deadline, that's stress, but that's not adversity. You don't face adversity just by going to a regular job. Adversity are particularly potentially traumatic events. They're not necessarily traumatic events in themselves, but there is potential for them to be traumatic. Sarah's going to be talking about adverse childhood experiences, or ACEs, a little bit later. The definition for those are these potentially traumatic events that occur in childhood, so prior to the age of 18. And then when we talk about trauma itself and a traumatic event, there has to be, so this is a definition from the DSM, the Diagnostic and Statistical Manual. This is what's used by clinicians to make actual diagnoses. And the definition in the DSM is exposure to actual or threatened death, serious injury, or sexual violence through one of these methods. So it can be actually directly experiencing it or witnessing it, learning that it's happened to somebody very close to you, or it could be repeated or extreme exposure to details of it. So the DSM has included this to incorporate first responders, but this is also relevant to people just having repeated exposure to potentially traumatic things over time. So I want to focus a little bit on this term threatened. Really, there's a level of subjectivity here. in terms of whether or not your life is actually threatened objectively doesn't matter so much as whether you think that your life is in danger. If you can imagine somebody pulling a gun on you versus somebody who is pointing their hand through their pocket, making it seem like they have a gun. The absence of an actual gun, the absence of that actual threat to life doesn't make it any less traumatic. There's a really interesting study that was done on London school children who saw news reports of 9/11 on TV. They're across the ocean. Their lives were not in direct threat. It was really just pictures on a screen, and they showed post-traumatic stress symptoms. So it's all about this perception of the level of threat. It's not about the objective threat itself. And just to come back to experiencing these repeated exposures of details of traumatic experiences or traumatic events, that sort of ties back to these early childhood experiences. You can imagine somebody growing up in an environment where they're repeatedly told about horrible traumatic things that are happening, either to people they care about or in the neighborhood. They don't necessarily have to witness it or experience it or have a loved one experience it. Those repeated exposures can create adverse experiences. So as I said, my background is in neuroscience. I'm going to talk a little bit about neurobiology and neurobiological response to trauma. There's something called the HPA axis, that stands for hypothalamic pituitary adrenal axis, which you can see on the right there. It's this, it's a system, it's a cascade of things that occur within the body when we experience stress. And it essentially activates the production of stress hormones like cortisol, which help us respond to those threats, or those stressors, I should say. And then I also want to mention the autonomic nervous system. So this is our nervous system that controls breathing and regulates our ability to interact with the environment. And I want to mention that there's connectivity with the limbic system early in development. I'm going to talk a little bit about the limbic system in a bit, but keep this in mind. So there is association between the autonomic nervous system and the limbic system. And we have this HPA axis that is activated when we experience a stressor. Trauma is experienced by pretty much everyone. There's an 80% likelihood of somebody experiencing trauma across their lifetime in the US. Doesn't mean you're going to develop PTSD, but when we do research on people with PTSD, they have the most robust traumatic symptomatology. So speaking to the effects of trauma, we speak a lot about cohorts of people who have been diagnosed with PTSD. So people who have been diagnosed with PTSD have been shown to have dysregulation of the HPA axis. So that's connecting to this HPA axis. If they experience multiple or severe traumatic events or early life events, traumatic events, then there is a shift in how the HPA axis actually functions, and that is lasting throughout the lifespan. And then people with PTSD also have low heart rate variability. So autonomic nervous system, heart rate, breathing. Heart rate variability is your ability for your heart rate to change based on the current circumstance. So the better your heart rate variability, the more adaptive you are to circumstances. And there's a reduction in this adaptability in people with PTSD. So their autonomic nervous system, connected with the limbic system, gets disrupted. We also see in everyone something called an orienting reflex. I'm sure that everyone is aware that trauma is emotional, and likely you've heard of emotional systems like the amygdala. People talk about the amygdala a lot, even lay folk, especially these days as we get more and more into talking about therapeutical interventions. So the amygdala is part of this limbic system that I mentioned that has connectivity with the autonomic nervous system. Yes, so here's the amygdala. You can see it here as well. This is an fMRI image of amygdala activation when people are experiencing emotion. So this is very robust data showing that the amygdala is associated with emotion. I'm going to talk about memory processes. Encoding and retrieval are both memory processes. This emotion center gets activated at multiple points in memory processes to bring our attention to emotional stimuli in the environment. And this is called the orienting reflex. It's particularly strong with negative stimuli, and it's so strong that it can override top-down modulation. So as humans, we have this massive prefrontal cortex. That's our reasoning center. We can say, okay, no, that shadow that just jumped out at me is my cat. It's not an intruder. You can downregulate your limbic system. But that immediate orienting reflex of, oh, there's a shadow in the corner, that immediate reflex overrides our prefrontal cortex's ability to downregulate. So again, talking about people in PTSD, they have both reduced medial prefrontal activity. So prefrontal cortex, that reasoning center, that's where you have this top-down modulation. So you see reduced activity in people with PTSD. So people who've experienced trauma are less able to downregulate those orienting reflexes, those big emotional responses, and also overall increased amygdala activity. So people who've experienced trauma have more hyperactive amygdala responses. Remember, amygdala is in the limbic system, and early in development, you get this connectivity between the autonomic nervous system, heart rate and breathing, with amygdala activity. So by experiencing trauma, your brain changes in such a way that it's also affecting your biological systems so that they respond to environmental stimuli in a different way. So shifting gears here a little bit, I want to talk about human memory. Most of my research has been in memory, and when it comes to trauma, PTSD is a memory disorder. And memory is a way for us to interact with our environment and optimize our own survival. It's based on what we've learned and what we remember that we can then successfully navigate our environment. So really, memory is a fundamental part of not only our survival, but also our responses to negative events, including traumatic events. I'm going to be talking about memory encoding. So consolidation and recall are later processes in memory. When you experience something, first it grabs your attention, then you encode it, which I'll talk about soon. And then there's a longer term process of putting it into long-term storage. That's called consolidation. And then when we actually remember an event, that's retrieval of that memory. Long before we're remembering events, our brains and bodies are already creating memories in a particular way that is going to shape the way that we interact with our environment later on. Part of this is that it's not enough to have that recall. We need to be able to react with immediacy when there's a potential threat in the environment. We can't sit around and think, oh, yes, when I was at this place at this time, this happened, maybe I shouldn't go here again. It has to be immediate. So this is why our bodily systems get tuned in conjunction with these events as they get stored into memory. So I said I'd talk about encoding. Encoding is essentially turning stimuli into a process or a construct that can be stored as later memory. When people think about this, they typically think about external stimuli, so information from the environment, right? There was a dog in the room, and my mother called me, and I saw a flash of light. All of that gets attached to this construct when it gets encoded, but so do internal stimuli. So things like your emotional state and the cognitions that you're having at the time, and bodily sensations. So if you have experienced a trauma in the past and you're already having a hyperactive autonomic nervous system, if you then experience something else, that hyperactivity of your autonomic nervous system, your dysregulation of your HPA axis, you will be feeling all of that in your body. And then that in turn gets added to this construct when you encode a particular event. So it's sort of a cyclical process. You're always using information from your past, from your present, and external and internal. Encoding is necessary for us to navigate our environment. It's very, very adaptive. So one, it reduces processing time. I'm sure everyone has heard about schemas. They factor into a lot of things, including bias, but they exist for a reason. We need to be able to process things really quickly, particularly in survival situations. So by attaching all of these different contextual stimuli to a particular memory, we can then reduce our processing time when we encounter similar things in the future. It helps us categorize our environment. It's particularly good at helping us detect threat. And one of the things, we also encode things based on relevance or irrelevance to our own life. So we're more likely to code things that are relevant to ourselves. And just as a note, there are individual differences in encoding processes. So this comes from past experiences, from temperament, from all kinds of things. But if I'm a trauma survivor and somebody else is not, the way that we encode new events is going to differ. So traumatic memory is a form of emotional memory, but it's not predicted solely by level of emotion or emotionality. All traumas are inherently emotional, but two different people can experience the same traumatic event, and one person might have a really maladaptive response and develop something like post-traumatic stress disorder, while the other person might go on to have a fully functional, healthy response. Part of that is because there are these differences in the encoding strategies that we utilize in how we interpret events. So in trauma literature, these are called paratraumatic appraisals. They're the primary predictor under our current understanding of trauma as to whether or not somebody's going to have a maladaptive response. So for example, develop PTSD and get a diagnosis of PTSD. And what it comes down to is how an event is processed cognitively as it occurs. So it's how you encode it, how you're using not just the external stimuli, right? So if two people experience the same trauma, they're experiencing the same external stimuli, but also these internal stimuli, bodily responses, cognitive encoding strategies. What that means is, what are you thinking about? How are you conceptualizing what's happening in reference to yourself, in reference to the world? That's a cognitive encoding strategy. Or in trauma literature, paratraumatic appraisals, para meaning as it happens. How are you appraising the trauma as it happens? And there are two primary paratraumatic appraisals that are predictive of trauma responses: threat to life and threat to self. I'm just going to bring all of these up. So on the left here, perceived threat to life. This is essentially in the definition of what a trauma is, as defined by the DSM. As I mentioned before, it is about perception. It's not necessarily about objective reality. In the severity of the trauma is unrelated to the objective risk of death. So you're bringing your past experiences into this event, and you're using those to gauge your current risk of death based on what you already know, what you've learned from the environment, what your body has learned. This is the primary predictor of severity of PTSD symptoms, and also a major predictor of post-traumatic stress symptoms in non-clinical individuals. So that means people who have not been diagnosed with PTSD. Everyone will experience trauma at some point throughout their life. Not everyone gets a PTSD diagnosis, but the maladaptive symptoms are predicted by whether they perceived a significant threat to life as they were experiencing the trauma. The other big one is threat to self. So our concept of ourself and the way in which we organize our memories and our self-concept and our identity is fundamental to our well-being. So even if you don't perceive a significant threat to your bodily well-being, if you experience something that threatens your self-concept and your identity, that is just as big a predictor as this perceived threat to life. People who have been through trauma or significant adversity, particularly a lot of childhood adversity, if that becomes the center of their identity, that becomes highly correlated with PTSD symptoms. People also who have been through significant trauma will have dysfunction in self-referent processing. There's disorganization in traumatic memory, and they're unable to actually process their sense of self to the same degree as people who don't have clinical traumatic symptoms. Back to these encoding strategies, perceived threat to life, threat to self. This sort of context that I just presented feels very negatively emotional. These findings remain true when people encounter neutral stimuli. If they are processing these as threatening to their life or threatening to their self, completely neutral event, they could have a traumatic reaction to that event. And when we explore some of these neutral encoding strategies, they suggest that they have adaptive origins. So when you encode neutral stimuli as relevant to your own survival, so this is sort of the flip side of threat to life. When you're processing things as a potential threat to life, it's also processing things as potentially advantageous for your survival. It's a dual process that you're doing simultaneously. It's bidirectional. There's recruitment of regions within the brain that are associated with human motor readiness, action preparedness, basically getting your body ready to, it's not quite fight or flight because that engages things like the HPA excess. You're going to have a surge of adrenaline, but this just primes your brain to be more attentive to things in the environment that could be relevant for your survival. And we see engagement of these neural regions in people when they're processing things based on one of these peritraumatic appraisals, irrespective of whether or not they're experiencing a trauma. All of that is to say, there is something inherently adaptive about the way that our bodies and brains respond to traumatic events and to adversity. Essentially, all of this makes us more aware of threat within our environment. It makes us more able to use our past experiences to respond to present experiences. And ultimately, if you are raised in an environment where you're surrounded by trauma, if you're in an abusive home, if you grow up in a war-torn country, being able to have a hyperactive response, emotional and physiological response to even neutral events that suggest threat that you, or remind you of threat that you've experienced in the past, that's adaptive. That's going to make it easier for you to navigate a traumatic environment. You want that to happen, and your body is designed to be able to do that for you. The problem occurs when you have experienced a lot of trauma, your body is tuned for a high trauma environment, and then you enter an environment that is safe. So you have these intense reactions, but there isn't any reason to have those reactions anymore. So there are solutions. Talking about that threat to self, regaining a sense of control is paramount in pretty much any intervention for adversity and trauma. This control in sense of both threat to self and threat to life. And we can retune our biological systems over time. So when we're talking about therapy, cognitive behavioral therapy, EMDR, any of that, this is over time retuning those biological systems that got tuned in a high traumatic event. And the limbic system, the amygdala, these emotional systems, they're involved not just in the engagement of traumatic stimuli, but also in the modulation of memory storage. So they're engaged at every stage of memory processing, and that opens an amazing door for reconsolidation therapy. So I said encoding is at the beginning, then consolidation, that's when your memory gets stored. When you retrieve a memory, imagine that your brain is a hard drive. You're basically deleting that file off of your hard drive, and you can do whatever you want to it before you save it, but that original file doesn't exist anymore. So when we retrieve a memory, the reconsolidation process makes that memory very labile. And reconsolidation therapy has been fantastic for people who've experienced trauma and adversity. So when they retrieve the memory, cognitive behavioral therapy, psychedelics like ketamine or MDMA have been amazing. Some other pharmacological interventions like propranolol, which is a heart medication. So that would be autonomic nervous system. You can have pharmacological interventions for any of these aspects that interrelate with trauma. And then in reconsolidation therapy, retune those physiological systems. And I won't get into this too much because this is Sarah's expertise, but there is some evidence of epigenetic interventions in reconsolidation therapy. So I do think I've used up a little more than my actual time. So I'm going to just speed things along here, do a quick summary, and then pass it to Sarah. Our brains and bodies are designed to adapt to our environment. Trauma responses are adaptive when your environment is filled with trauma and adversity. You want your brain and body to react in that way because that keeps you alive. The more severe the trauma or the more occurrences you have, or whether you experience trauma during neural development, i.e. childhood, those all lead to more severe and more lasting traumatic symptomatology. So it's harder to retune your body. There is a difference between, dysfunction and adaptation only due to the environment that you're in. And keep in mind that cognition and emotion are inseparable, and they're inseparable from the body as well. All of these work together. It's not just brain or body, brain and body. It's how you think, it's how you feel, and it's how you function. And I'm going to now pass it to Sarah, who is going to talk about biological embedding, which is essentially all of this.
Thank you so much, Taylor. That was fantastic. And it's so important to understand that we have all of these biological processes happening simultaneously, and realistically, that there are consequences biologically for that occurring. And one of the theories of how this happens is called biological embedding. So what is biological embedding? It's a theory that our lived experiences can get under our skin. And by that, we mean can potentially affect lifelong changes in our biological processes, like what Tayler was talking about. So, for example, exposure to a traumatic event may lead to conditionally adaptive biological changes. For example, your autonomic nervous system or hypervigilant attention that allows for detecting these threats as soon as possible, which, while helpful in the context of trauma and therefore adaptive in that context, may be maladaptive or dysregulated for later health functioning in a different context. However, these biological adaptations, they can lead to durable alterations by shifting the pathways of our homeostatic processes. So what does that mean? That means that these dysregulated biological responses, like differences in heart rate variability, HPA axis dysregulation, increased amygdala activity, hyperattentiveness, all these things that Tayler just talked about, they can act as mechanisms through which early life experiences can then affect our later health and development. So, for example, if we look at adverse childhood experiences, or ACEs, which include instances of abuse and neglect, they can produce these toxic stress, but also these physiological symptoms that can then affect, as we just heard, multiple brain regions and physiologies. And this can in turn make people more vulnerable to health problems across the lifespan. So, for example, an increasing number of adverse childhood events has been robustly associated with later life differences in health. And these are all different examples of that. So, for example, self-report of having poor physical health or emotional health, more likely reports of chronic pain, more likely to have lung and asthma problems. And additionally, recent research has shown that children of color are systemically exposed to disproportionately high levels of adversity and consequently associated with more health problems. One such biological process that may act as a link between these experiences is epigenetics. So what is epigenetics? Epigenetics are persistent and potentially lifelong alterations in your genomic information that do not alter your underlying DNA sequence. So while your gene sequence never changes throughout your whole life, the resulting biology and health differences that you can see can potentially change through epigenetic pathways. And social epigenetics is the process by which our life experiences can affect these biological pathways, which in turn influence our epigenetic pathways and result in different biological outcomes in a very cyclical manner. Notice Tayler and I are talking a lot about the cyclical relationship between our experiences and our biology. So while there are multiple epigenetic modifications, today we'll be discussing DNA methylation. So this is the deposition of a methyl group onto your genome at specific sites. So one way to think of this is to consider the genotype that you're born with and that does not change as this window of potential expression. And that could be understood as, for example, the wattage of a light bulb. And the brightness of this light is the expression of your gene. In this case, DNA methylation then acts as a dimmer switch, adapting the expression of your genes within the limitations of your genotype as influenced by the environment. For example, experiences of childhood adversity and trauma. So the classic example of DNA methylation and changes in phenotype is something called the agouti mouse. So these are all agouti mice. All of these mice are genetically identical and offspring of different mothers. So while the mothers were pregnant, they were fed varying amounts of methyl-rich food, and that resulted in similarly varying amounts of methylation in the agouti gene of their offspring. These offspring were all fed the same diet, they were all environmentally treated the exact same way, and yet they have very different coat colors and increased rates of obesity, diabetes, and cancer due to the diet that was fed to their mothers. So while these two mice are in every other way identical, the methylation of the agouti gene results in these stark phenotypic
differences.
Identical twins are another example of how our biology can change, even when starting with the same genotype. Here are two famous identical twins as babies, as adolescents, and as adults. As you can see, while they still look very similar, as time goes on, they will start to look less and less alike, as will their epigenomes. While there are many biological features of DNA methylation, one particularly interesting and useful aspect is that the methylation value at specific sites in the epigenome change reliably over time, consistently across individuals. So as we can see in this figure, as each of these three individuals grow older, the dimmer switch of DNA methylation is acting the same way in the same parts of the genome. So in this way, DNA methylation at these sites allows us to be able to calculate an individual's biological age. This is what's known as the epigenetic clock. From the epigenetic clock, we're able to determine epigenetic age, which is an age, your biological age, that represents the functioning, growth, and health of a person at a point in time. So we see here an example in blue of an individual who is epigenetically young for their age, because they exhibit phenotypes much later into their chronological life associated with aging. Whereas the person in red is someone who is epigenetically old for their age. They exhibit aging phenotypes much earlier than the person in blue. These differences of biological age from chronological age is known as epigenetic age acceleration, or how fast a person is aging relative to their chronological age, as depicted in this graph, with the red aging epigenetically faster, the blue aging epigenetically slower, and the dashed line showing what concordant aging would look like. Research in adult cohorts has provided robust evidence that accelerated biological age is associated with increased morbidity and earlier mortality. And adversity appears to associate with faster epigenetic aging. For example, there's substantial evidence that the experience of trauma and adversity will accelerate your epigenetic aging and associate with risk for future health difficulties, such as the epigenetic age of children with confirmed cases of child maltreatment who are aged between 2 and 9 years old, who had been legally removed from the care of their biological parents by child protection services and sheltered in residential care facilities. And we see here on the left that the children on the left, who are these children who have been removed, their epigenetic age is significantly higher than children from the same age from the same community. Additionally, age acceleration was also associated with children's direct experience of violence and decreased heart rate. So children who were predicted to be older than their chronological age had twice as much violence exposure as other children, and their heart rates were actually very similar, not to that of children, but to that of adults. As we can see, the epigenetic clock has been robustly associated with these early life exposures. In recent literature, increases in epigenetic age acceleration have been consistently associated with experiences of stress and adversity, most especially experiences of abuse and maltreatment, but also including poverty, hardship, and living in disadvantaged neighborhoods. These associations with childhood adversity have also been found while in pediatric age acceleration, which we see here, in adolescent epigenetic age acceleration, and even in retrospective accounts of adult and older adult methylation. So though there is a consistent association with the experiences of childhood trauma and adversity and increased epigenetic aging, it's still unclear what the developmental ramifications of this are. However, the health ramifications of this in older adults do appear to associate with worse health and faster aging. So knowing that these experiences of adversity are biologically embedded, can these also last into the next generation and be passed on to our children? And this brings us to the question of intergenerational trauma. Intergenerational trauma is the topic of much research today, and it is the transmission of an individual's experiences and/or historical oppression and their negative consequences across generations. There's evidence of the impact of intergenerational trauma on health and well-being, as well as the health and social disparities facing many populations. It is possible that different parental environmental exposures can influence inherited phenotypes in offspring. Environmental differences experienced by fathers, such as changes in diet, stress, or chemical exposures, may transmit non-genetically inherited phenotypes to their offspring. This includes altered metabolism, behavioral changes, or modulated susceptibility to stress or chemical exposures. For example, in humans, a recent study found that the children of fathers who smoked cigarettes before puberty, approximately before the age of 12, were much more likely to have higher BMIs and difficulty with their metabolism. There are several considerations that must be made, though, when we're talking about intergenerational or transgenerational trauma and transgenerational epigenetics. First is the discussion of social transmission versus biological inheritance. So while these are both ways for parents to essentially transmit experiences, either trauma or adversity or oppression to their children, they have very different mechanisms. The first being social transmission, which is through environments that the parent creates, their relationships. They are then transmitting the experiences that they had, because these are things, as Tayler talks about, that are encoded into our memory, consolidated into our memory, and affect the way that we experience our world and the way that our world and our children experience us. Biological inheritance, on the other hand, happens when gametes, so the sperm or the egg, are biologically, physically different, and they are marked specifically by an event. And so for social transmission, for example, in animal models, if you have cross-fostering or IVF, which means that the biological parent is not the parent who has raised them, you would not see the social transmission. However, for biological inheritance, IVF and cross-fostering wouldn't make a difference, right? For the intergenerational social transmission of trauma, there is certainly evidence for this, specifically around parental experiences, both early life trauma and adulthood trauma, that in turn affects the social environment of their children. So some examples of this would be, the cycle of poverty, secondhand exposure to chemicals like smoke, especially tobacco, has been probably the most well-studied. Disrupted sleep schedules, both parents and subsequently children's disrupted sleep schedules that can sometimes happen. Other types of availability of quality time and parenting style, and mental health difficulties that can affect all of these aspects. Another thing to keep in mind is also the type of biological inheritance. As there are two that we can talk about today, mitotic and generational. So mitotic heritability is acquired heritability. This is passed from cell to cell, which means that the cells in your body that are affected by an environment and acquire, for example, a DNA methylation mark in infancy could still be there in adulthood, but that is only within a single individual. This type of mitotic heritability is something that we see in caterpillars to butterflies. It is through this process of DNA methylation change that that metamorphosis happens. But it isn't passed on to another generation. It is within a single individual. Intergenerational or transgenerational heritability is passed from parent to child, and this is inherited from past generations. We also want to talk about the DOHAD hypothesis, or the developmental origins of health and disease versus heritability. So this actually refers to effects that happened during pregnancy and the experience of the environment of a pregnant mom. And so rapidly growing fetuses and neonates, right, are very vulnerable, for example, to nutritional differences. These are not transgenerational or intergenerational effects because they're affecting mom and the fetus, but transgenerational inheritance would be if an actual biological mark was transferred rather than having an environment that affected both the parent and the fetus at the same time. So an example of this would be the Dutch hunger winter famine, which in November 1944, the World War II Axis blockaded against the Dutch, and thousands of people starved. Children who were born from pregnancies during that time were born with different methylation patterns. Specifically, they had less methylation in an insulin-related gene. And this was caused by the mother's diet, specifically starvation, while pregnant. And then these same children in adulthood had difficulty with metabolism, glucose, BMI, and obesity. Another important factor is also whether we are talking about maternal or paternal generations. So for epigenetic changes in mammals that can arise and can be induced by the environment, there are differences between whether or not we are looking at a female line or a male line. Because if in a female, in this case, mouse, in this example, she was pregnant with a fetus, that can also be affected in utero, as are the eggs of that fetus that are also in utero, because female mammals are born with their eggs. So it actually has to go 3 generations before we can even think about transgenerational effects. This explains why the majority of the work in this field has been done with male mice, because it's actually 1 generation less before you can show transgenerational inheritance. And it's much easier to test sperm than it is eggs. However, there are, using these male mouse models, evidence for epigenetic transmission of transgenerational inheritance. The best evidence that we have is from these mouse models, specifically with learned behaviors from traumatic events that can be passed on from the father mouse to their offspring. A male mouse was behaviorally conditioned to associate a specific smell that is only detected by mice, only at 1 receptor that's encoded by 1 gene, which is why this is a really good study. And this is the M71R gene. The subsequent startle response to the smell, because they are anticipating a shock, was then passed on to that male mouse's offspring, both through traditional mating and through IVF, even though those mice had never been conditioned. The male mouse never interacted with any of the offspring, and therefore could not teach the response, and the mice must have inherited that response. They were born with the startle response to the smell. These mice had an increase in the receptors for perceiving that smell as well, and the male mouse's sperm had methylated that DNA, the DNA of that particular gene for that particular receptor, as did his offspring, and therefore that offspring's offspring. While simple, these mouse models are the best evidence that we have that learned behaviors can actually be epigenetically inferred. However, the actual mechanism by which this occurs is unknown, as DNA methylation is erased during the development of gametes. This is called epigenetic reprogramming. We know that it happens, and we know that the DNA methylation is erased, so we don't actually know the direct mechanism through which this could occur, which is why this is slightly controversial. Currently, the best hypothesis we have is that the transmission of paternally environmental changed inherited phenotypes that could be happening through epigenetic information is actually encoded through these things called small regulatory RNAs. These are microRNAs have been identified as potential carriers of this epigenetic information, and that they will come in the sperm. Again, this has only really been looked at in males to then reconstitute these DNA methylation patterns in later generations. However, this is still currently unknown how this works. This is the current hypothesis of how this could be taking place. However, it's really important to note that DNA methylation is still changeable. While there are sensitive periods of greater change and potentially intergenerational transmission, though we don't know the mechanism how yet, DNA methylations can be removed or added throughout life. So for example, smoking can clearly be seen in DNA methylation patterns of a single gene, the AHRR gene. And while those who have never smoked have the highest DNA methylation, former smokers are much more like those who have never smoked than those who are currently smoking. And we can see the less one has smoked, the more like never smoking these people are. So it illustrates that though smoking may be visible in your genes, so is quitting smoking. It's important to know this because the intergenerational transmission of trauma, regardless of how it occurs, is not a fixed or predestined or inescapable thing. Instead, while, for example, DNA methylation does change with time, it is also possible that intervention can mitigate risk or perhaps even promote future resilience. So, for example, many clinicians and scientists have created interventions to address the experiences of trauma, for example, trauma in children. And though we live in an imperfect world, and it would be great to exclude all experiences of trauma, as Tayler said, 80% of people will experience some level of trauma in their life, some more extreme than others. And if we can ameliorate these effects for children who have already experienced it, that is our goal. So one example of this is child-parent psychotherapy, or CPP, which is an intervention model for young children under the age of 5 who experience at least one traumatic event and or have post-traumatic stress disorder. This treatment is based on attachment theory and integrates lots of different other types of psychodynamic and developmental theories and methods. These sessions include child and parent together or primary caregiver, and the goal of CPP is to support and strengthen the relationship between that child and their caregiver as a vehicle for restoring the child's cognitive, behavioral, social functioning, and sense of control in the world. Treatment also focuses on contextual factors that may affect the relationship and is particularly culturally sensitive. So at multiple sites and with this wonderful research team, we asked the question, would CPP demonstrate biological improvement at the DNA methylation level from this intervention? And this intervention has been found to be very effective, especially at treating PTSD symptoms, where children who had experienced trauma and had this particular intervention did see a significant decrease in the symptomology of their PTSD. And so in our study, when we looked at baseline of children with high amounts of adversity who did and did not participate in this intervention, their epigenetic age acceleration was the same. There was no difference. But after the participation in this intervention, there is, or an equivalent amount of time in the people who did not receive it, there is a difference between the two groups. Specifically, those children who were matched but did not receive the intervention increased their rate of epigenetic aging as opposed to the children who were intervened on. And these results indicate that not only does adversity accelerate epigenetic aging, but intervention can mitigate that acceleration and prevent future further biological aging. And even without intervention, just being in a supportive family environment can mitigate epigenetic age acceleration from adversity, such as experiences of discrimination. So here we can see in 2 different cohorts that even in high discrimination situations for racial minority adolescents, epigenetic age acceleration does not increase for those with supportive family environments, but does increase for teens without family support. So together, we hope that this work serves to promote a framework of biological adaptation to intervention, and in doing so, promote agency to support change in environmental contexts from the bottom up and the top down. So thank you very much to everyone who supported this work, and especially thank you to the Alliance Against Violence and Adversity and everyone who made this talk possible today. And thank you all for coming.
So thank you everyone for attending, and thank you so much, Sarah and Tayler, for being such amazing knowledge sharers today. We really appreciate you.
Thank you so much for having us, and thank you everyone for coming.
Thanks so much.
Yeah, it was a pleasure to speak for you.
Thanks for joining us on the Her Health podcast, transforming health for women and girls at risk. Remember, your health matters, and together we can make a difference. Until next time, remember to prioritize your health and well-being. For more resources and support on issues affecting girls, women, and gender-diverse people, visit our website at avatraining.ca. This is Dr. Nicole Letourneau. Thank you for listening.